Good morning. It's October 28th, and this is your daily brief in science. Here's everything you need to know. In a remarkable achievement for high school students, Kelsea Johnson and Nikia Jackson have proven the Pythagorean theorem using trigonometry, a feat once considered impossible. Their groundbreaking work has been published in the American Mathematical Monthly on October 28, 2023, featuring a total of 10 proofs, 9 of which are entirely new. Motivated by a $500 prize, Johnson and Jackson dedicated significant time and effort to their project, overcoming various challenges along the way. They are now only the third and fourth individuals to successfully prove the theorem using trigonometry without falling into circular reasoning, marking a significant milestone in the field. Their initial proof utilized the law of sines, effectively avoiding the circular reasoning that has historically hindered trigonometric proofs of the theorem. The Pythagorean theorem itself posits that the sum of the squares of a right triangle's two shorter sides equals the square of the hypotenuse. Jackson expressed hope that their achievement will inspire other students to persevere through challenges and recognize their potential in mathematics. Both students have emphasized the importance of representation for young women and women of color in STEM fields, showcasing their pride in this accomplishment. Their work received validation through peer review and was presented at an American Mathematical Society meeting earlier in 2023. Their hard work earned them keys to the city of New Orleans and personal praise from former First Lady Michelle Obama. Mathematician Alvaro Lozano Robledo commended their contributions, underscoring that high school students can indeed make significant impacts in mathematics. Currently, Johnson is studying environmental engineering at Louisiana State University, while Jackson is pursuing a doctoral degree in pharmacy at Xavier University of Louisiana. Breast cancer continues to be a pressing global health concern, with 2.3 million new cases and 685,000 deaths reported in 2020. This stark reality highlights the urgent need for advancements in early detection methods. In response to the limitations of traditional mammography, researchers are developing a sensitive blood test aimed at improving early breast cancer detection. Utilizing advanced liquid chromatography coupled with high-resolution and tandem mass spectrometry, the study identified a 20-lipid panel that achieved an impressive area under the curve of 0.95. This panel exhibited a sensitivity of 0.91 and specificity of 0.79, demonstrating its potential effectiveness. The research successfully distinguished breast cancer patients from healthy controls by identifying 23 lipids in extracellular vesicles, resulting in an overall accuracy of 0.82 and sensitivity of 0.85. Machine learning techniques played a crucial role in enhancing the predictive accuracy of these lipid signatures. An ensemble model outperformed simpler models, achieving a stable accuracy of 86.1% and high sensitivity in detecting breast cancer. This indicates significant potential for clinical application in early diagnosis. The study also developed a multivariate plasma-derived lipid biomarker signature from 598 blood samples, effectively differentiating healthy individuals from those with breast cancer. Extracellular vesicles in blood are rich in tumor-specific analytes, making them valuable for diagnostic purposes. The methodology demonstrated high accuracy and reproducibility, positioning it well for future clinical applications in biomarker validation. Additionally, understanding variability in plasma protein levels due to factors such as sex, age, and ethnicity is essential for accurate disease diagnosis. Comparative analysis between European and North American cohorts revealed significant differences in 42 proteins, suggesting varying physiological pathways in disease development. The findings emphasize the potential of specific proteins as key indicators for diseases, advocating for region-specific diagnostic panels. Esophageal cancer is a pressing global health concern, ranking as the 7th leading cause of cancer-related deaths worldwide. Among its various forms, esophageal squamous cell carcinoma, or ESCC, accounts for approximately 85% of new cases. Recent research has shed light on the role of cancer-associated fibroblasts, or CAFS, in the progression of ESCC, showing how they enhance tumorigenesis through their interactions with tumor cells. Adipose-derived stem cells, known as ASCS, can transform into CAFS when exposed to signals from tumors. This transformation increases their secretion of extracellular matrix components and pro-inflammatory cytokines, which are crucial in supporting tumor growth. The metabolic reprogramming of both ASCS and CAFS is essential for their tumor-promoting activities, with a notable shift towards glycolysis that fuels tumor development. The study emphasizes the intricate relationship between CAFS and cancer cells, particularly highlighting the role of Amphiregulin, or AREG, in ESCC progression. Bioinformatics analyses reveal a significant increase in AREG expression in ESCC compared to normal tissues, suggesting it could serve as a potential therapeutic target. Treatment with AREG has been shown to improve the survival and migration of ESCC cells by activating the EGFR, ERK/P38 MAPK signaling pathway. Key signaling pathways, including TGF, beta, WNT/beta-catenin, and Notch, play vital roles in regulating epithelial mesenchymal transition, or EMT, and CAF activation. EMT is a critical process in cancer progression, allowing tumor cells to invade surrounding tissues and metastasize to distant organs. Potential therapeutic strategies for ESCC include disrupting interactions between ASCS, CAFS, and tumor cells, targeting key signaling pathways, and exploring options in immunotherapy. Emerging research also suggests that the gut microbiome may influence the behavior of ASCS and CAFS, with microbial metabolites potentially modulating their differentiation and function within the tumor microenvironment. Additionally, previous studies indicate that co-culturing human bone marrow-derived mesenchymal stem cells with ESCC cell lines can enhance malignancy in cancer cells. A recent study has unveiled promising insights into the cytoprotective effects of hydrolysates derived from the red Californian worm, Eisenia fetida. Researchers focused on how these hydrolysates can mitigate oxidative stress in differentiated CACO2 cells, a type of human intestinal cell line. The findings confirmed that at the tested concentrations, these hydrolysates do not exhibit cytotoxic effects, maintaining 100% cell viability. The study suggests that low molecular weight peptides from Eisenia fetida could offer therapeutic benefits for conditions related to oxidative stress, highlighting their potential as a source of bioactive peptides. Out of the 28 peptides identified in the F less than 3 kilodalton fraction, 5 showed antioxidant activity, with Kepidwater demonstrating the highest affinity for key proteins myeloperoxidase and KEAP1. Molecular docking techniques were utilized to explore the interactions between these peptides and target proteins, further elucidating their antioxidant mechanisms. The F less than 3 kilodalton fraction exhibited significant cytoprotective activity, enhancing cell viability by 50% against H2O2-induced oxidative stress and restoring redox status. Oxidative stress occurs when pro-oxidants exceed antioxidants, leading to cellular damage linked to health issues such as cancer and cardiovascular diseases. In addition to the worm study, the research also optimized the extraction of crude polysaccharides from the edible mushroom Agroscibe agarita, assessing their antioxidant and anti-aging effects. The polysaccharides demonstrated an IC50 value for superoxide anion scavenging at 4.693 mg per milliliter and improved the lifespan and locomotion abilities of the model organism, Caenorhabditis elegans, indicating potential anti-aging effects. The study employed response surface methodology to optimize extraction parameters, revealing significant interactions among solid-to-liquid ratio, extraction temperature, and time. The optimal extraction conditions were established with a solid-to-liquid ratio, yielding a concentration of 0.034 grams per milliliter, an extraction temperature of 92.64 degrees Celsius, and an extraction time of 2.82 hours, resulting in a yield of 11.325% plus or minus 0.996%. Diabetes mellitus is a prevalent metabolic disorder, making the regulation of carbohydrate-digesting enzymes like alpha-glucosidase crucial for managing blood glucose levels. Type 2 diabetes, characterized by insulin resistance and beta cell dysfunction, underscores the need for enhanced strategies to control postprandial hyperglycemia. Current anti-diabetic medications that inhibit alpha-amylase and alpha-glucosidase often come with side effects, prompting researchers to explore safer, natural alternatives. A recent study focused on the inhibitory mechanism of polyphenol-rich rose extracts against alpha-glucosidase, utilizing multispectral and molecular docking techniques. The findings revealed a significant inhibitory effect on alpha-glucosidase activity with an IC50 value of 1.96 micrograms per milliliter, improving to 1.33 micrograms per milliliter following ultrasound pretreatment. Thermodynamic analysis indicated that hydrogen bonding plays a key role in the interaction between the rose extracts and alpha-glucosidase, resulting in a spontaneous reaction. Fluorescence spectroscopy demonstrated that the rose extracts can quench the intrinsic fluorescence of alpha-glucosidase, suggesting a strong interaction. The Lineweaver-Burke assay further indicated that these extracts function as mixed-type inhibitors of alpha-glucosidase, impacting the enzyme's conformation and microenvironment. Importantly, the non-cytotoxic nature of the extracts, combined with their ability to reduce oxidative stress in cell models, supports their potential role in diabetes management. Additionally, Asphodylus microcarpus contains bioactive flavonoids that may inhibit enzymes and exhibit antioxidant and antimycotic properties, making it a promising natural source for managing type 2 diabetes. Ethanolic extracts from various parts of Asphodylus microcarpus significantly inhibit alpha-glucosidase, with IC50 values up to 25 times lower than a carbose, while demonstrating low alpha-amylase activity. The extracts also exhibited strong antioxidant properties and low cytotoxicity, likely attributed to the presence of phenolic compounds. This has been your Daily Brief in Science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.